Good morning, guys, or afternoon. I always say that because it's morning when I record these. Whatever, nobody cares. So, today we're going to talk about our last macromolecule, which is nucleic acids. So we've already talked about carbohydrates that provide structure and function. We've talked about lipids, which provide structure and function. Oh my gosh, structure and energy. Wow, not enough coffee. Maybe I can talk now. And then we've talked about proteins, which provide many, many functions. I think that's where my brain was going in your body, including making up enzymes and making your blood cells able to carry oxygen, making transport molecules, all of these different things that we're going to talk about more heavily when we talk about individual cells. But now we are talking about nucleic acids. We're not going to get into genetics until like a couple units from now. So we're mainly just kind of like introducing nucleic acids, refreshing your memory on them, updating some of the things that you know about their structure. So when we're looking at nucleic acids, their function is to store hereditary information. Okay, so purely genetic code. As far as we have two main groups of them, we have DNA and RNA. So DNA is our double-stranded helix. We have A, G, C, and thymine, A, G, C, and T. Very, very large molecule, and part of a nucleic acid is actually a sugar monosaccharide called deoxyribose. Deoxy, D means without oxys, oxygen. So it's actually a ribose sugar that is missing an oxygen group. If we look at our other one, we have RNA, which is single-stranded, and it has the bases AGC and uracil. So adenine, guanine, cytosine, and uracil. This carries information from DNA to ribosomes that are also made of RNA. We have a couple different types as well. We have tRNA, which is transfer RNA. That's the one that actually brings amino acids to the ribosomes. We have rRNA, which is what ribosomes are made of. mRNA, that's our messenger that goes between the nucleus and the cytoplasm where the ribosomes are. And then RNAi, which is actually like a defense system for our RNA in our body and for getting rid of viruses and different things like that. When the RNAi sees DNA in your cells that doesn't belong to you, chops it up and it kills it to get rid of it. And the sugar in this one is ribose. So if we look more closely at the actual structure of our nucleotide, we have our phosphate group. So we know that is very polar. We have a 5-carbon sugar. So first carbon is up here, 2, 3, 4, 5. And then we have an oxygen at that apex. And then up here in the blue, we have a nitrogenous base. The reason why we call it a nitrogenous base is because it looks like that carbon ring, but we have nitrogen mixed in with it. So it's not purely a sugar or a carbohydrate. So it's a nitrogenous base. This base is the part that gets changed out if it is an adenine, a guanine, a cytosine, a thymine, or a uracil, depending on if it's DNA or RNA. That's the only part that changes, along with this pentose sugar as well, if it's RNA or DNA. So if we look really, really closely at these bases, here we have the pyrimidines. The only difference between a pyrimidine and a purine is a pyrimidine is a single ring and a purine is a double ring. Okay, so a single ring, double ring, single ring, double ring. So cytosine, thymine, and uracil, and we see nitrogenous base. We see the nitrogen groups in there. I see an amine group. I see a methyl group. I see a C double bond to an O. So that is either our aldehyde or a ketone, depending on where it is. Down below, we have our purines, which are double ring. Okay. Over on the side, we have our sugars, deoxyribose versus ribose. So down below here, we can see that ribose has an extra hydroxyl group, whereas deoxyribose does not have that hydroxyl group. Okay, so that's the difference between them. So if we look back at our other picture on the last slide, this would actually be DNA because it's missing that second hydroxyl group. Okay. So once again, going ahead, the only difference in these sugars is that O missing. Okay. When we're looking at the way that these carbons are numbered, we're going to go counterclockwise around our circle. So we have 1, 2, 3, 4, and 5. This becomes really, really important when we talk about DNA structure because if you remember, the DNA strands aren't parallel, they are anti-parallel. So one strand goes in one direction and one strand goes in the other direction. And the way we actually denote that is with these carbons. So if a strand is going from 5 prime, we see up here, to 3 prime, it's facing up. And if it's going from 3 prime to 5 prime, it's facing down. So you can imagine that whole little blue thing. Ooh, let's see if I can do some movie magic. Can I move this around in here? No, it's not letting me. Arr. Now I'm mad. Okay, but if you flip the whole little thing around, facing the other way, and yeah, here's a picture of that. So if we're looking at our groups on each side here, we have our sugar phosphate backbone. So we have a phosphate group that's polar, and we got a sugar and a phosphate and a sugar and a phosphate and a sugar. The bond between these is the phosphodiester bond, the phosphodiester bond. Phosphate connecting with an oxygen group, so that would be a phosphodiester bond. And we can see that on this side, the 5 prime end, or that number 5 sugar is up, or the number 5 carbon, I'm sorry. On the other side, that 3, according to that backbone, is the one that's facing up along that backbone. So we call that the 3 prime end. When we're also looking at how these things are put together, a pyrimidine or a 1 ring is always going to link up with a purine or a 2 ring. The reason why is because if we have a ladder and you have a long rung and a little short rung, that's going to match together. On the step below, you also need a long rung and a short rung. So they are even on each side, because if you have 2 shorts, then your ladder sides are going to go in. And if you have 2 longs, then your ladder sides are going to bow out. So we need a short and a long to always match up with each other. And if we can look at this out as well, we see in the middle, we have hydrogen bonds happening. So here our cytosine has 3 places that can actually bond only to guanine, because guanine also has only 3 places to bond. If we look at thymine and adenine, those have to go together because of 2 places to bond and 2 places to bond to make that hydrogen bond. So once again, just looking at this structure, we have a double ring. So that's going to be a purine with a pyrimidine. I know that this is guanine because I see 3 hydrogen bonds. G always matches with C, so I know that this is cytosine because I see it's bonding with 3 bonds and it's a 1 ring. Down below, I see that it's bonding with 2 hydrogen bonds, so I know that this one is an adenine, and I know that this one is a thymine. When we call these things a double helix, what we actually mean is, yes, the DNA is twisted, and then the twist is twisted. That's what we mean by a double helix. So it's not the fact that it's 2 sides that are twisted, it's that the twist is twisted. When we're looking at this, you can see it really, really well in this picture where we have a minor groove and a major groove. Okay, not super, super important, but this is why we call it a double helix, because it's a twist within a twist. And we have a little minor part and a major part, and the way that all goes together is just because of how everything hydrogen bonds and phosphodiester links together. If we're following the flow of genetic information, we're going to go from DNA to RNA to protein. We call this the central dogma of biology. So that word dogma means like a central belief system. So just for an example, if we're talking about like a major world religion, Buddhism, more people are Buddhist than they are Catholic in the world, their central dogma is that Buddha is their like God and brings in part reincarnation. If we're talking about the central dogma of Catholicism, we're talking about the Father, Son, the Holy Spirit. If we're talking about the central dogma of Judaism, it's that there is one God, and it's kind of the Old Testament stuff. All of that is a dogma or a central belief system. So the central belief system of biology is the fact that DNA gets converted into RNA. That RNA then gets read by ribosomes to get transferred into proteins that we talked about the other day in order to do different functions. So in eukaryotes, DNA is always located in the nucleus, and that information cannot leave. DNA is very, very fragile, and if it leaves the nucleus, we have a possibility of it getting destroyed. So that DNA is going to stay within that nucleus. The way the message gets out is that mRNA is going to copy just the genes that we need. So those things get turned on and off. We'll talk about that later in the year. And then that RNA can leave and get read by the ribosome to make our polypeptide, puts together those peptide bonds to make our protein. So looking at the different types of RNA that we have ribosomal RNA or rRNA. So this is actually how proteins get made. And we can see that this actually, it looks like a protein, like it looks like the pictures that we saw the other day, but it is in fact just strands of these single pieces of RNA all looped together that act and do a very, very specific job. If we look at transfer RNA or tRNA, this is what actually decodes that information. So down below on the tRNA is actually an open spot that has an anticodon that will read 3 bases at a time. That anticodon is linked up here to a specific amino acid. So what happens is that tRNA will come down on the ribosome. It will read that codon and anticodon, just like we did with like the codon charts in regular bio. And then that amino acid is up on the top, so it knows which one to connect. These tRNAs are reusable. So as soon as it gives up its amino acid to put into that protein link, it will go around and find another amino acid that specifically links up to it. So the functional group is actually the part that links to the tRNA. It's very, very specific. So it's going to link together and find another one and then bring it back. So these are reusable. The last kind is typically the kind people think of when they think of RNA, and that's our messenger RNA or mRNA. And that's going to carry the genetic information from the nucleus to that ribosome site in order to do protein synthesis. So that is really all of the notes for nucleic acids at this point. We're going to go way more into them and how all this gene control stuff works later when we talk about genetics. So that's really it for right now.